In my presentation today, what I have shown is it's very important to understand the mechanisms of development, how the brain is built, and with that information then it is possible to implement strategies to stem cells in order to promote their differentiation. So the two main messages of my presentation were that one family of molecules which are called WNTs, and these are morphogens. They work during development and they sculpture the phenotype of the cell, how the cells look like at the end, depending on their dose. Now there is a lot of work in this area, and we know that which are the specific doses of these factors and which is the combination of these factors, WNTs, together with other factors, for instance like sonic hedgehog. Now this specific combination of factors is actually what determines what fate a cell will take, so how will the cell look like at the end. So it's not a single factor, it's a combination of different factors at different doses, and then during development at different times you have a different pulses of those signals. So it's the factors themselves, the dose, and the temporal aspect, so how long they are exposed to those specific signals. So this is how morphogens work. So in this case what we found is that WNT1 and WNT5A, two of those morphogens, are very important for the development of dopamine neurons, and we need to use them in order to make good preparations of dopamine cells. In my second part of the presentation, what I looked is at nuclear receptors, which are transcription factors that regulate also gene and development function. In this case what I reported is on the function of ligands that bind to these receptors and activate transcription. And our findings indicate that these ligands are cholesterol derivatives, so that's basic metabolism from our own body. And these unusual molecules are actually, we found that they have a very important function in the generation of neurons. So now with this knowledge, what we have used is combine WNTs and combine treatment with cholesterol derivatives, and what we can now make is improve preparations of dopamine neurons. Now these dopamine neurons show a character that is very similar to those that we find normally in the brain, and what we are doing now with these cells is using them for cell transplantation in animal models of Parkinson's disease, and at the same time we are using them for drug discovery. Basically, we have a very good cell preparation in a petri dish. We can expose those cells to small molecules, to different drugs, and develop new medicines for Parkinson's disease. Since those cells that we are making are actually human cells, and we can actually use human cells derived from Parkinson patients through the iPS technology, then we can actually model disease in vitro with a human dopamine cell and expose those cells to small molecules and test their potential. So the advances in our presentations in the session today, what make possible is to generate cells in vitro in a petri dish that could be used for screening by pharmaceutical industry or by investigators, and identify then new molecules that could be useful in the future for Parkinson's disease. Now both of these two therapies that I mentioned, cell transplantation and drug discovery, are two options that are for the future. But we now have the tools, and we believe that we are getting closer to develop novel therapies for Parkinson's disease patients.